Okay, guys, we can get going. First and foremost, thank you so much to everyone that's on the call. It's really great to see all of you that have joined. And just to introduce myself to anyone who's unfamiliar with me or with who Assay Genie is, my name is David and I'm the distributor manager here at Assay Genie, and I manage our distributor portfolio. And today I'll be talking you through therapeutic antibodies, anti-drug antibodies and biosimilars, and innovations in detection and monitoring of these technologies. And throughout this talk today, I'll be speaking to you about the quantitative, qualitative, and free drug versus total drug ELISA kits that we have in our portfolio. And I want to introduce you to someone. And I want you to meet Sarah, who is a patient and a person who is living with rheumatoid arthritis. And Sarah has just started a biologic therapy like adalimumab, but her symptoms return after 6 months. So, why does this matter? Well, this matters because some patients respond well, but others experience loss of response to the drug, and side effects or total treatment failure is common. Reasons include that drug levels drop and there are not enough drugs in circulation in the body to work. And another reason is that the body fights back and the immune system creates anti-drug antibodies that neutralizes the therapy. And this shows that it's actually very important and very critical to actually monitor the drug and the measurement of ADAs, which is very important for drug efficacy assessment. And this is how the workflow in Sarah's assessment would begin. It would initially start with measuring the free drug levels, where the physician or her doctor will quantify the therapeutic antibody levels. Next, they would assess the anti-drug antibodies, and if drug levels are low, they would then test for the presence of anti-drug antibodies with ELISAs to evaluate the immunogenicity. Next, they would evaluate the neutralizing antibodies, and if anti-drug antibodies are detected, this would confirm whether neutralizing antibodies are impacting the drug efficacy. Finally, their physician or doctor would then optimize or adjust their therapy, and then based on the results, they would adjust the dosage, modify treatment intervals, or switch biologics as needed. This is just the agenda for today, where I'll speak to you about who we are as a company, why monitoring therapeutic monoclonal antibodies and biosimilars are important. I'll give you an overview of therapeutic antibodies, both the past and the present situation on them. I'll speak to you about our solution. I'll also discuss how we assess therapeutic antibodies, and I will also show you some future perspectives and market analysis. And I'll finish this presentation and this talk with a summary of what we discussed, and then I'll also go to a Q&A session. So if it's your first time here at Assay Genie, you're very welcome. And if you're unfamiliar with our story, this is our history. Assay Genie was founded back in 2017 by our co-founders, Dr. Colm Ryan and Dr. Sean McFarrig. Assay Genie is headquartered in London, but our primary base of operations where we QC all of our products is in Dublin, in Ireland. We have distributors in over 80 countries and over 1,000 product citations, and all of our products are ISO 9001 certified. And on a daily basis, we work with the world's largest pharmaceutical companies, including Pfizer, Lilly, and Johnson & Johnson. And on a daily basis, we are collaborating with the world's best academic institutions, including Oxford and Cambridge University and Harvard University on top of that. And this is our company's mission, which we strive to and adhere to every single day that we're working, which is to make scientists smile through using our innovative products, our fun marketing, and our remarkable interactions with our team and with our partners and customers around the world. And if you do not know anything about Assay Genie's technology platform, this is a great place to start. Assay Genie focuses on 5 key technologies in our portfolio. We have over 32,000 validated ELISA kits in our portfolio. We have an amazing collection of different multiplex arrays and membrane arrays where you can detect anywhere from 1 to 274 proteins. This rivals our competitor Luminex. We have over 11,000 highly pure recombinant proteins in our portfolio and a wide range of different biochemical, activity, and cell-based assays. And last but not least, we have over 40,000 validated antibodies in our portfolio too. So I want to pose a question to everyone in the audience today, which is why should we monitor therapeutic monoclonal antibodies and biosimilars? And I'll answer this question using a few case studies and challenges. So to begin with, we want to monitor therapeutic monoclonal antibodies and biosimilars to ensure efficacy and safety in the patient. We can monitor therapeutic antibody levels to optimize treatment within the patient. It's also critical to monitor these to track pharmacokinetics, to measure the drug concentration over time in biological samples. And it's also very important for dose optimization, where this helps to adjust the dosage to maximize the benefits and minimize toxicity to the patient. Why it's also very important to do is because it's critical for biosimilarity evaluation, where you can test the similarities between biosimilars and the reference products. And our solution at Assay Genie is that we provide over 70 different ELISA kits for the therapeutic drug detection and monitoring. We offer these ELISA kits for specific diseases such as asthma, where we have ELISA kits that can monitor therapeutic drugs such as infliximab and omalizumab. We also have then therapeutic drug monitoring kits for other diseases such as osteoporosis, where you can monitor the levels of drugs such as denosumab. And then for diseases such as arthritis, where we have ELISA kits, you can monitor the concentration of adalimumab and then certolizumab. And on top of that, we do have ELISA kits that can monitor the concentration of drugs used in cancer treatment, such as cetuximab and ipilimumab, and also nivolumab. But included in this special webinar is our ELISA kits that can quantify therapeutic antibodies, that can quantify biosimilars, and that can quantify anti-drug antibodies too. And all of these ELISA kits can be used to monitor these specific proteins and antibodies, both in serum and plasma samples. And these are available for human, mouse, non-human primate species, and other species too. And it is also worth noting that we do also provide a custom ELISA service for the development of specific therapeutic antibody monitoring kits. And I want to share with you a success story where a specific lab group wanted to understand the pharmacokinetics and clinical outcomes of nivolumab. And here, this research group from the regional medical research center in India. This is the lab group that did it. And when they were actually using our nivolumab human ELISA kit, they wanted to measure the low dose of nivolumab in patients with advanced cancer. We supplied this ELISA kit for the quantification of the drug in patient plasma samples, and we support them by providing the validation data and recommending specific dilutions. They concluded that low-dose nivolumab leads to lower exposure in patients as compared with conventional dose, but low dose was better tolerated. And another success story I wanted to share with you was from a lab group from the University of Baghdad in Iraq, where they were investigating the correlation between polymorphisms and natalizumab response in MS patients. And here, they wanted to assess the occurrence of antibodies against natalizumab treated multiple sclerosis patients with polymorphisms on the ITGA4 gene. We supplied the natalizumab antibody screen and ELISA kit for serum samples from patients, And we supported them by providing the validation data and recommending specific dilutions. The paper concluded that 3 polymorphisms had significant effect on the propensity to be a natalizumab non-responder in Iraqi MS patients. And now I want to actually just speak to you a little bit about therapeutic antibodies, what has happened in the past with them, and where we currently stand with them in the present tense. And if you joined us for our biosimilar webinar that we launched last month, you might have seen this similar timeline of therapeutic monoclonal antibodies. And here we just really discuss and talk about the developmental history of monoclonal antibodies from mouse to human origin. And the reason why I'm repeating this figure and showing it in today's talk is because it just shows how quickly therapeutic monoclonal antibodies have advanced from the lab bench to the patient's bedside. And we can see that the first cancer vaccine was developed back in 1890 by William J. Coley. And then in 1975, we had the birth of hybridoma technology. Then in 1978, we had the discovery of tumor-specific monoclonal antibodies. And in 85, we had the first study done with adoptive T cell transfer in cancer patients. Then in 1990s, we had the seminal discovery of a checkpoint inhibitor by Professor James Allison and by Professor Tsuko Honjo, who were co-awarded the Nobel Prize in Physiology and Medicine for this discovery. Then in 1997, transgenic mice were used for generating fully human monoclonal antibodies. And then in the early 2000s, in 2002, we had the first FDA-approved humanized monoclonal antibody, rituximab. And then in 2006, we had the first FDA-approved humanized monoclonal antibody being used for solid tumor treatment in patients. But before we go any further, I just want to give out some simple definitions as to what therapeutic antibodies are, what biosimilars and anti-drug antibodies are, because some people can usually get a little bit kind of mixed up with what each of these are. So therapeutic antibodies recognize specific proteins on cells to initiate an immune response and are used to treat cancer, arthritis, and other diseases. An example of this would be CTLA-4, and then the treatment therapeutic antibody for this would be ipilimumab, and the treatment therapeutic antibody for PD-1 will be either nivolumab or pembrolizumab. And then with biosimilars, these are antibodies that are almost identical to their therapeutic counterparts. An example here would be that Pfizer demonstrated therapeutic equivalence to an EU-sourced reference adalimumab in trials. And lastly, anti-drug antibodies are antibodies that are generated by the immune system to clear therapeutic antibodies. These can actually have a negative effect on treatment, so monitoring ADA levels is very important after drug treatment. And these are the types of therapeutic antibodies currently in the market. And these therapeutic antibodies have their limitations. To begin with, I just want to give you guys a definition of what immunogenicity is. And immunogenicity is the ability of a foreign substance to provoke an immune response. And when we look at the monoclonal antibodies right here, we can see the range of potential for immunogenicity. We can see that both murine and then chimeric antibodies have a high to medium high chance to provoke an immune response, whereas humanized or human antibodies have a low chance of generating an immune response. And monoclonal antibodies do have their own limitations and challenges that impact their quality and that impact their effectiveness. To begin with, some of their limitations include their stability, where monoclonal antibodies can unfold partially or completely denature, causing their aggregation. Another limitation would be post-translational modifications, where both glycosylation and oxidation can lead to reduced efficacy and increased immunogenicity. And then a specific challenge that they still really need to address is that they have a high percentage of remaining in the bloodstream and cannot penetrate cancer tumors effectively. But these limitations have led researchers to produce more advanced antibody therapeutics, or next generation antibodies as they're called. These include bispecific antibodies, antibody drug conjugates, FC engineered antibodies, and also nanobodies too. And there are a number of different categories of therapeutic antibodies on the market used in cancer treatment. The first one is naked monoclonal antibodies, and these can induce cell death by different mechanisms, including antibody-dependent cellular cytotoxicity and also by complement-mediated cytotoxicity, which direct the targeting of cancer cells to the tumor microenvironment. But on top of that, we do also have checkpoint inhibitors or immune checkpoint blockades. The second category are bispecific antibodies, and these target two receptors and are engineered to further enhance therapeutic effects. The third one that I think everyone has heard about and that is commonly in the media or in the news for recent advances in these therapies are CAR T-cell antibodies. And this involves inserting the gene for a chimeric T-cell receptor antibody where it targets a specific cancer marker into T cells, and this targets and then kills the specific cancer cell. Last but not least, we then have our conjugated monoclonal antibodies, and these are antibodies carrying different molecules to the tumor site and can kill cancer cells. So included here, you have your antibody drug conjugate, you have your radioimmunotherapy conjugated antibody with your radioligand, you then have your immunotoxin conjugated antibody, and then you have your small interferon RNA particle conjugated antibody. So there's a wide range of different therapeutic antibodies to choose from. And I want to just briefly discuss how anti-drug antibodies are generated in our bodies. To begin with, at the first stage is the biotherapeutic recognition stage. And here, after administration, the drug is actually endocytosed by the antigen presenting cell, which initiates T cell activation. And in this immune cell activation stage, you have activated T cells which release cytokines and stimulate naive B cells to recognize the biotherapeutic antigen, and which becomes activated. In the final antibody production stage, activated B cells differentiate into plasma cells which produce anti-drug antibodies targeting the biotherapeutic. But this then raises the question of why should we monitor anti-drug antibodies? Well, monitoring ADAs is very important for immunogenicity and for efficacy assessment, where therapeutic monoclonal antibodies can induce an immune response, leading to ADAs productions, and high levels of ADAs may neutralize the therapeutic effects of the drug. Secondly, it's very important to monitor ADAs for clinical response variability, where some patients may develop ADAs that impact drug levels and clearance. It's important to monitor them for safety concerns too, as ADAs can potentially lead to adverse events, including hypersensitivity reactions. And lastly, it's very important for biosimilarity evaluation, and monitoring ADAs helps evaluate the immunogenicity profile of biosimilars compared to the reference product. Now, I just want to actually give you an example of why it's very important to monitor ADAs. And I think a perfect example to use is the recent rise of Ozempic use around the world. And for those of you who are unfamiliar with Ozempic, otherwise branded by Semaglutide or Wegovy, this is a weight loss drug that has helped people living with obesity drastically lose weight in conjunction with working out and eating well. And GLP-1 impacts a wide range of different organ systems in our body, where in the brain it may decrease appetite and increase satiety. In the kidney, it would increase natriuresis. In our fatty tissue, it increases lipolysis and increases glucose uptake, whereas in the pancreas, this could increase insulin secretion and decrease glucagon secretion. In our GI tract, it decreases gastric emptying and acid secretion, and in our muscles, it increases glycogen synthesis and glucose oxidation. And in the heart, it has a wide range of varying effects, including decrease in blood pressure, increase in heart rate and myocardial contractility, and also increase in diastolic function. So studying and monitoring the ADAs against GLP-1 can actually help with our understanding of treatment effectiveness. It can help us with understanding the pharmacokinetic and the pharmacodynamic variability, and can also help us in assessing the long-term impact of GLP-1 or Ozempic on the body too, and is also critical for investigating any kind of side effects, and can help us develop specific neutralizing antibodies. And I wanted to give another example too of how we can actually monitor the anti-drug antibodies for other compounds. Another one of these compounds is called polyethylene glycol, and polyethylene glycol, otherwise called PEG, was generally classified as generally recognized as safe in foods. But a polyethylene glycolated lipid is used as an excipient in both the Moderna and the Pfizer-BioNTech vaccines for SARS-CoV-2. And reports show that PEG is immunogenic in animals and in humans, and both anti-PEG antibodies exert significant influence on an in vivo performance of polyethylene glycolated therapeutics. This compromises the efficacy of polyethylene glycolated therapeutics. So what's the problem here? Begin with, detection of therapeutic antibodies and biosimilars currently requires outdated and expensive techniques such as HPLC. Secondly, monitoring of ADAs is crucial to maximize drug efficacy and dosing strategies, and kits for their detection are limited and not quantitative. Lastly, competitively priced kits are difficult to find for therapeutic monoclonal antibodies, biosimilars, and anti-drug antibodies too. Our solution at Assay Genie is that we have a wide selection of therapeutic antibodies, biosimilar, and ADA ELISA kits, all of which are ISO 13485 certified. We have highly validated ELISA kits for free versus total, quantitative, semi-quantitative, and qualitative measurement of therapeutic antibodies, biosimilars, and anti-drug antibodies. And last, but certainly not least, we have premium PhD scientific support, which is available via live chat, email, phone, or video to support experimental setup, instrument optimization, and data analysis too. Now, I'm going to just speak to you about how we can assess therapeutic antibodies, and I'll just discuss with you some validated ELISA kits for antibody screening and quantification. And these are some of the top global therapeutic monoclonal antibodies, which includes pembrolizumab, adalimumab, daratumumab, nivolumab, and also then pertuzumab. And at Assay Genie, we offer unique ELISA kits for the quantitative, semi-quantitative, and qualitative of total drug, free versus total, and anti-drug antibodies in human, mouse, and non-human primate and other species for both serum, plasma, and cell samples too. And we also offer anti-polyethylene glycol antibody ELISA kits. We offer 12 in total. Specifically, we have 5 anti-PEG IgG ELISA kits for human, monkey, mouse, rat, and rabbit species. But then we have 7 anti-PEG IgM ELISA kits for human species, monkey, mouse, rat, rabbit, and for canine samples too. And these kits are unique as they offer the quantitative indirect measurement of these compounds, and they are also highly sensitive to the nanogram per milliliter range, and these have been extensively validated in the serum, plasma, and cell culture samples. And this is the overview of the protocol, which has been optimized and can be used effectively for determination of monoclonal antibodies or anti-drug antibodies. It's very easily adaptable and can be used both in serum or plasma samples, and it only requires only 10 microliters of sample and can be used with all common reagents in the lab. Here, we prepare the sample and the reagents. You add the standard samples or blanks to the wells. You follow this over with the incubation and the washing steps. You allow the color develop, and then you read the results on a plate reader, and then you can plot the results and calculate concentration. It's very straightforward. So, why should anyone choose our TDM ELISA kits? As I always say before, our kits are extensively validated, and there's a wide range of kits to choose from, which are very easy to use. Here, I'm just giving 2 examples of our TDM ELISA kits, including our adalimumab ELISA kit and also our etanercept ELISA kits. And you can see here that the standard curves have been generated for both of these targets, where the absorbance is just mapped against the concentration. It's very straightforward and very easy to follow these ELISA kit protocols. And lastly, why anyone should choose our ADA ELISA kits is because these are extremely unique, because they can be used to quantify both the free and total ADAs using acid dissociation. And in our ELISA kits, we add a weak acid to the sample, which dissociates the drug antibody complex. And once the sample is neutralized, the ADA can then bind to the biotinylated drug, forming a bridge and allowing the detection of the ADA. This is very important and key for immunogenicity testing. And our competitors do not use acid dissociation technology, meaning they are not giving a complete and relevant and thorough ADA ELISA kit. So our kits allow researchers to make informed decisions when they are quantifying these targets. And here, when you just look at this figure both in purple and in green, you can see here that adalimumab is bound by ADAs against it, making it difficult to detect the full amount of anti-drug antibodies. But the acid dissociation frees it from the ADAs, allowing for its complete and full detection here in green. And if you want to integrate one of Assay Genie's TDM and ADA ELISA kits into a preclinical trial, this is how you do it. If you're looking to measure free drug levels, you can use Assay Genie's Therapeutic Drug Monitoring ELISA kit or Biosimilar ELISA to quantify the therapeutic antibody levels. Then, when you're looking to assess anti-drug antibody levels, and if these drug levels are low, you want to test for the ADA presence with ELISA to evaluate the immunogenicity. Next, you want to evaluate the neutralizing antibodies, and if ADA is detected, confirm whether neutralizing antibodies are impacting and affecting the drug efficacy. Lastly, you want to optimize and adjust the therapy, and just like with our patient Sarah, based on the results, you want to adjust the dosage, modify treatment intervals, or switch biologics as needed. And just to give you guys some future perspectives and a little bit of market analysis on where we see therapeutic antibodies going in the future, we can see that the market size and share will be increasing over the next 4 to 5 years. And we can see through a compound annual growth rate that this will increase by around 14.5%. And you can see here, these were the figures back in 2022 and 2023, and these are the projected figures for 2029. And the market share by research shows that cancer will dominate the global antibody drug market, and then autoimmune diseases is very much expected to grow at the fastest rate too. And these are some of the top antibody drug manufacturers currently around the world, which includes Lilly, Pfizer, AstraZeneca, Regeneron, and AbbVie. So, just to really summarize everything that we talked about today, increasing interest in the development of new therapeutics for cancer and autoimmune diseases is going to be crucial for developing the latest therapies around the world. drug monitoring and measurement of ADAs is crucial for drug efficacy assessment. And assessment of long-term effects of therapeutics, such as Ozempic or Semaglutide, is also key to understand their impact on a patient's health. And lastly, Assay Genie offers a wide range of different ELISA kits for both the detection and monitoring of drugs for advancements in translational research and preclinical studies. So thank you so much to everyone who listened to this too. It was great that all of you could join. I'm very happy to take any questions or queries that anyone might have. So please feel free to turn on your mic or put the questions into the group chat. If no one has any questions, you can please feel free to send me an email and I will answer any of your questions then too. You can type in. Oh, no worries at all, Mirta. It might be something just with the settings. If you just want to maybe type in your question and then I can see on working on the settings. Yeah, so that's a great question there from Samira. So Samira's question is, if there's anti-drug antibodies generated against a drug, what is generally the next step? Stop the drug or neutralize the antibodies? I think it usually depends. I think Samira did want to normally just like neutralize the antibodies, or they could stop the drug too. I think usually they'd maybe assess the situation and see what is the best course of action. But if the patient's life is in danger, then they would normally want to actually stop the drug immediately just to prioritize the patient's health. Assay Genie solutions are used on radioligand therapies. Mirta, that's a great question. I'd need a little bit of further expansion on that. If you want, we can follow up on an email and I can give you further information on that too. But I don't have all of the information ready for you right now to kind of provide a specific answer to that. Okay. Yeah, that's great. I can absolutely follow up with an email in relation to that. But yeah, I wish I had the answer for you right now for that, but I will work on getting for you too as well. So I'll touch base with you after this, and we can find a solution together. Any other questions, guys? Can you also briefly pitch the key questions to ask a customer to sell the ADA solutions? Yeah, absolutely. So when you're working with these customers, a lot of these customers will be working with a lot of patient samples too. I think honestly, one of the first questions I appreciate the honesty, Mita. I appreciate the honesty. Look, I think number one, when you're looking for these customers, you want to basically first start off by asking them, are they working with patients? Are they working with anti-drug antibodies and biosimilars? Because obviously these are going to be in a very unique field. So you want to make sure that they are, number one, working with these. And then you want to also ask about, like, what kind of drugs they are working with. And then because we have a number of different therapeutic drug monitoring ELISA kits, we have over 70 in our portfolio. Once you begin to find out what drugs they are working with and with what patient samples, then you can begin to narrow it down then to our therapeutic drug monitoring ELISA kits too. So find out drug, find out sample, and then also find out really what ways that they are doing it too, and then mention that we have a number of anti-drug antibody TDM ELISA kits, biosimilar ELISA kits in our portfolio that will be able to measure it too. So not every single lab is going to be doing this. This is going to be a unique cohort of researchers out there. So you will need to narrow your search field. But once you have that search field narrowed and you have these customers known, if you're able to sell them our ELISA kit, they will generally be repeat customers. That is what we have found. Once you actually are able to kind of recognize them, they will then come back and actually buy more of them too, because it is such a unique field they're working in. I hope that helps. No problem. Any other questions, guys? I don't think there is. So if anyone does have any further questions, remember you can always reach me on distributors@assaygenie.com, and I will follow up to you as soon as possible. I will send out this recording today along with just the email and the contents of it and what was just talked about today. So if you want to, and if you can, please share this with your customers, and please discuss this with your customers. And remember, we're always on hand to answer any specific questions or queries that you have. So no question or query is too big or too small for us to look after. Thank you again for all of you for taking the time out of your schedule to join me on this call today. And I hope you all have a great rest of your afternoon. And yeah, I'll be speaking to you again sometime soon. Thanks so much, everyone. I am going to end this meeting now, so take care.